and we will present about glucose oxidase. This is our group member. For the task 1, I will be present about the general information about glucose oxidase, the enzyme classification of glucose 1.1.34, and glucose oxidase need a cofactor to assist the activity. The similarities of glucose oxidase found in living organism of various family genus. We can find glucose oxidase from Aspergillus niger and Penicillium. Both catalyze the oxidation of B-glucose to glucose 1,5-lactone. The differences of GOX found in living organism of various family is GOX from Penicillium is less stable compared to the Aspergillus niger, but has higher affinity for BD-glucose and higher specificity constant to Aspergillus niger. For this, the catalyzed reaction for glucose oxidase. Next, for task 2, the glucose oxidase is important in the application of industrial. First, its catalytic product, which is hydrogen peroxide, with antibacterial and antifungal agent. Glucose oxidase also used in food preservatives, baking, dry egg powder production, wine production, and gluconic acid. Next, its electrochemical activity, important component in glucose sensor. And lastly, monitor glucose level in fermentation, bioreactor, raw material, and food product.
Okay, so how do we measure the enzymatic activity? What protocol do we use? So we use modified Lowry protein assay. So as a beginning of every experiment, you should begin with 3 triplicates. So you need to have your own sample and also control. So for the sample, you should mix it with buffer, and you should make it into 400 microliter per dilution. And just same for the sample, which BSA and mix with buffer, and you do it the same, which is 400 microliter per dilution. So, and then you start with the experiment. First of all, you use Lowry concentrate, and then you mix it and you incubate it in room temperature, and incubate it for 10 minutes. And then after incubation, you mix it with following reagent, and you should mix it. You should add the following reagent, and you should vortex it very quickly. And then you incubate it for 30 minutes, and then you could take the solution and count the absorbance at 750 nanometer. And so, why do we use modified Lowry protein assay? So as you know, if Lowry protein assay, they can only withstand under low temperatures, but modified, they can withstand in room temperature. And this we can compare with Kumasi-based assays, because Kumasi-based assays, they could withstand in room temperature. So this gives us an advantage. And also, Kumasi-based assays, they are very acidic. So for proteins who are acidic intolerance, they could not undergo Kumasi-based assays. And Lowry protein assays demonstrate less protein-to-protein variability compared to Kumasi-based assays, which shows 30% more variability.
Okay, how do you get from Aspergillus niger cell, and you extract from the asparagine laser that contains the GLX. So first of all, the asparagine laser cell, you obtain it and then you suspended it in 50 ml of destruction solution. Then after that, after this destruction, you destruct it again mechanically. Then inside 4 degree 15 minutes. After that, since the homogeneous is produced, part of the homogenate will that consists the cell debris will be frozen at negative 20 degrees Celsius for direct enzyme extraction. Then the remaining portion will undergo centrifuge and the supernatant is collected and frozen. Next.
So how do we purify GOX? So first we have to set the conditions, temperature, precipitation, centrifuge at which speed, and dialysis step for how many hours. Okay, first 50 grams of aspergillus niger is suspended into sodium acetate buffer and dialyzed against sodium acetate buffer. Next, it's centrifuge to remove brown precipitate. The supernatant is applied on amber light column, which has been buffered with sodium acetate solution. Next, the column is washed with 4 liters of sodium acetate buffer, and the enzyme that has been eluted as yellow solution and sodium acetate buffer is added ammonium sulfate to 90% saturation until a protein is precipitate. After it has been centrifuged, the precipitate has redissolved in sodium phosphate buffer and dialyzed against sodium phosphate buffer. Then it is applied on DEA E-cellulose column and buffered against sodium phosphate buffer. The protein form is brown in color and it's washed against phosphate buffer until a yellow protein is eluted. The precipitate is redissolved in water and dialyzed against water and kept in deep freeze.
Okay, so now we are going to demonstrate on how what are the alternatives that we have to demonstrate that the protein that we have is purified. So one of the alternatives is mass spectrometry, because mass spectrometry, they could separate atoms, isotopes or molecules based on mass. But one of the problems here is that what if there are other proteins who have the same molecular mass as glucose oxidase? So, but then if you can see here, they are divided into 3 parts, which is first the sample will be ionized, which positive charge will pass through to the electromagnet, and it will be detected if the positive charge was on top of the electromagnet, it means it's heavy. If it's lower, it means it's light. So next is by SDS page. SDS page, like usual, as presented as other groups as well. You need to put some buffer, which is trace buffer, and you put your sample and it will be loaded. So the third option that we have is Western blotting. So in order to do Western blotting, you need to do sample preparation first, and then you need to do SDS page, and also you need to do antibody probe. As mentioned by Eka just now, when the protein will bind to the membrane, So they use first primary antibody will bind to the protein that we want, and then followed by secondary protein which has enzyme, and they undergo chromogenic reaction where it will precipitate its substrate so that we can detect our particular enzyme. So next we have is salt bridge. Salt bridge is just maintaining the enzyme in a neutral conditions. We want to ensure that it is in neutral condition, so that we use salt bridge because salt bridge is just basic electron transfer between, like we know, is copper and zinc. Next, we can also use DLS, which is dynamic light scattering device. This device is created based on the principle of Brownian principle. He's a botanist who was investigating a pollen where it shows pollen they scattered very random. So based on this principle, they create DLS. So DLS ensures, it doesn't ensures, it prevents protein aggregation by light scattering devices, because the light will pass through the sample and it will be diffracted, and the light diffracted will pass through another lens, and it will go to the correlator, and it will be presented on the PC, which is PC. Okay. So how do we demonstrate that there are no significant contaminants? One of the ways is, first of all, as we all know, is agarose gel. But another alternative that we have is ICP-MS, which is inductively correlated plasma mass spectrometry. It applies mass spectrometry processes. It's just that, first of all, argon, the sampler, it uses auto samplers, and it will be transferred into the nebulizer with argon, and they will release aerosol, and this will be ionized and followed by mass spectrometry processes, and it will be processed in the computer as well.
Okay, next is how to determine the 3D structure of glucose oxidase. First, we have to go to the PDB database and search for glucose oxidase. Then click on glucose oxidase from Aspergillus niger. Download the file in PDB viewer, and you have to have Yasara database. Load the file and you can view the 3D structure of glucose oxidase. These are the 3 active sites found in glucose oxidase. Okay, then to obtain the active site of an enzyme without any lab work is to go to UniProt database and to compare the sequences of the amino acids with other species. So here is the sequence obtained.
I'm going to make it quick. Lastly, how to determine the protein structure. First, we use X-ray crystallography. we can visualize the protein structure, and this is easy because the protein can be seen in atomic detail, very detailed, and we can see the whole 3D structure. Okay. Okay. Next, we use nuclear magnetic resonance (MNR). The information can be obtained about the structure and dynamic of a protein. Basically, this is the molecular parameters can be examined more, and all the data can be used by a computer apparatus to calculate the whole 3D structure. Last is electron microscopy. One form is the transmission electron microscopy, and it has a powerful magnification, produce high resolution and two-dimensional images, and produce information of surface feature, shape, size, and structure. These can determine their chemical bonds and also the disorders of the protein structure. Okay, that is from us. Thank you very much.